Welcome back to Anatomiqa. In the last session, we completed discussing naming of the voluntary muscles, blood supply, and nerve supply of the muscle. In this session, we will discuss on the details of the histology of muscular tissue. The learning objectives of the session are: mention the types of muscle tissue, describe the features of skeletal, cardiac, and smooth muscles, differentiate between the microstructure of skeletal, cardiac, and smooth muscles, describe the connective tissue framework of the muscles, explain the functional and clinical correlation. Muscle tissue is composed of cells that optimize the universal cell property of contractility. As in all cells, microfilaments and associated proteins generate the forces necessary for the muscle contraction, which drives movement within organ systems of blood and of the body as a whole. Essentially, all muscle cells are of mesodermal in origin and differentiate by a gradual process of cell lengthening with abundant synthesis of the myofibrillar proteins actin and myosin. They are called muscle fibers since cells are elongated. Muscle specialists refer to certain muscle cell organelles with special names. The cytoplasm of muscle cells is often called sarcoplasm. The smooth endoplasmic reticulum is the sarcoplasmic reticulum, and the muscle cell membrane and its external lamina are the sarcolemma. The variation in diameter depends on specific muscle, age, gender, nutritional status, and physical training of the individual. Exercise enlarges the skeletal musculature by stimulating formation of new myofibrils and growth in the diameter of individual muscle fiber. This process, characterized by increased cell volume, is called hypertrophy. Increase in number is hyperplasia, which takes place very readily in smooth muscle whose cells have not lost the capacity to divide by mitosis. Three types of muscle tissue can be distinguished on the basis of morphologic, functional characteristics, with the structure of each adapted to its physiologic role. Skeletal muscle contains bundles of very long cylindrical multinucleated cells with cross striations. Their contraction is quick, forceful, and usually under voluntary control. Cardiac muscle also has cross striations and is composed of elongated, often branched cells bound to one another at structures called intercalated discs that are unique to cardiac muscle. Contraction is involuntary, vigorous, and rhythmic. Smooth muscle consists of collections of fusiform cells that lack striations and have slow involuntary contractions. In all types of muscles, contraction is caused by the sliding interaction of thick myosin filaments along thin actin filaments. The forces necessary for sliding are generated by other proteins affecting the weak interactions in the bridges between actin and myosin. Let us discuss in detail about the structure of all the three muscles. Let us start with skeletal muscle. Skeletal or striated muscle consists of muscle fibers which are long cylindrical multinucleated cells with diameters of 10 to 100 micron meter. During embryonic muscle development, mesenchymal fibroblasts fuse where the cell membranes of individual cells fuse together to form a single membrane, leading to myotube with many nuclei. These myotubes then further differentiate to form striated muscle fibers. Elongated nuclei are found peripherally just under the sarcolemma, a characteristic nuclear location unique to skeletal muscle fibers or cells. A small population of reserve progenitor cells called muscle satellite cells remains adjacent to most fibers of differentiated skeletal muscle. Skeletal muscles help in quick and forceful contraction. Now let us see how is the organization of a skeletal muscle. Thin layers of connective tissue surround and organize the contractile fibers in all three types of muscle, and these layers are seen particularly well in skeletal muscle. The entire muscle is surrounded by an external sheath of dense connective tissue called epimysium. Septa from the epimysium extend inward, carrying the larger nerves, blood vessels, and lymphatics of the muscle. This thin layer of connective tissue sheath forms the perimysium surrounding the bundles of muscle fibers called the muscle fascicles. Each fascicle made of group of muscle fibers forms a functional unit in which the fibers work together. Nerves, blood vessels, and lymphatics extending along the perimysium supply each fascicle. Within each fascicle, external lamina of individual muscle fibers is surrounded by a very thin, delicate layer of reticular fibers and scattered fibroblasts called the endomysium. In addition to nerve fibers, capillaries form a rich network in the endomysium, bringing oxygen to the muscle fibers. Collagen in these connective tissue layers of muscle serve to transmit the mechanical forces generated by the contracting muscle cells. Individual muscle fibers seldom extend from one end of a muscle to the other. Some skeletal muscle fibers taper at the ends where the epimysium is continuous with the dense regular connective tissue of a tendon at myotendinous junctions. Ultrastructural studies show that in these transitional regions, collagen fibers from the tendon insert themselves among muscle fibers and associate directly with complex infoldings of sarcolemma. Here you can see the animation of the whole muscle formed by several bundles of muscle fibers. Each bundle or fascicle is formed by a group of muscle fibers. Each muscle fiber contains numerous myofibrils and each myofibril consisting of myofilaments made of actin and myosin. In the previous few slides, we discussed the organization of the muscle. Now we will discuss on organization within muscle fibers. Each skeletal muscle fiber is made of bundle of myofibrils. Each fiber, when longitudinally sectioned, show cross striations of alternating light and dark bands. The dark bands are called A bands, that is anisotropic, or birefringent in polarized light microscopy. The light bands are called I bands, that is isotropic, which do not alter polarized light. Each I band is bisected by a dark transverse line, the Z disc. The part extending from one Z disc to another Z disc, which is the repetitive functional subunit of the contractile apparatus, is called the sarcomere and is about 2.5 micron meter long in resting muscle. The muscle fiber is enlarged to show the details. The sarcolemma of the muscle fiber is cut and reflected to show the inside of the muscle fiber. The sarcoplasm of each muscle fiber contains primarily long cylindrical filament bundles called myofibrils, which have a diameter of 1 to 2 micron meter and smooth endoplasmic reticulum called sarcoplasmic reticulum. Myofibrils are unbranched contractile units running parallel to the long axis of the muscle fiber. Mitochondria and sarcoplasmic reticulum are found between the myofibrils. Myofibrils consists of an end-to-end repetitive arrangement of sarcomeres through its length. The lateral registration of sarcomeres in adjacent myofibrils causes the entire muscle fiber to exhibit a characteristic pattern of transverse striations. The drawings show the inside of the individual muscle fiber on the left side, and on the right side the ultrastructure of the arrangement of myofilaments, which can be observed only under the electron microscope. Each myofibril consists of longitudinally oriented protein filaments called myofilaments. Myofilaments are of two types, thick and thin filaments. The A and I banding pattern in sarcomeres are due mainly to the regular arrangement of these two types of myofilaments. The thick myofilament is composed of proteins myosin and thin filaments is composed of protein actin. These are organized within each myofibril in a symmetric pattern containing thousands of each filament type. The thick myosin filaments are 1.6 micron meter long and 15 nanometer wide. The middle non-overlapping and the peripheral overlapping parts of the myosin filaments form the A band at the mid region of the sarcomere. So A band present in the center of sarcomere contains thick filaments and part of thin filaments overlapping with each other. Close observation of the A band shows the presence of a lighter zone in its center corresponding to a region with only the rod-like portions of the myosin molecule and no thin filaments, called the H zone or H band. Bisecting the H zone is the line containing a myosin binding protein, myomycin, that holds the two thick filaments in place and creatine kinase. This line is called the M line. The thin helical actin filaments are 1 micrometer long and 8 nanometer wide and run between the peripheral part of thick filaments. The non-overlapping part of the thin filaments form the I band. The center of I band, where the actin filaments from adjacent sarcomeres meet, is called the Z disc or Z line or crosses membrane. Actin filaments are anchored perpendicularly to it. The actin filaments exhibit opposite polarity on each side of this disc. In a cross section, one myosin filament is surrounded by 6 actin filaments. Thin filaments, apart from actin, are also tightly associated with 2 regulatory proteins: tropomyosin, located in the group between the 2 twisted actin strands; troponin, which is a complex of 3 subunits; troponin T, which attaches to tropomyosin; troponin C, which binds calcium; and troponin I, which regulates the actin-myosin interaction. To provide uniform contraction of all muscle fibers, the skeletal muscle possesses a system of transverse tubules or T-tubules. In skeletal muscle fibers, the smooth endoplasmic reticulum or sarcoplasmic reticulum is specialized for calcium sequestration. To trigger calcium release from sarcoplasmic reticulum throughout the fiber simultaneously and cause uniform contraction of all myofibrils, the sarcolemma is folded into a system of finger-like invaginations called transverse or T-tubules. These long finger-like invaginations of the cell membrane penetrate deeply into the sarcoplasm and encircle every myofibril near the junction of A and I band boundaries of sarcomeres. Adjacent to each side of every T-tubule, the sarcoplasmic reticulum is expanded to form terminal cisternae. In longitudinal sections, this complex of a T-tubule with 2 closely associated terminal cisterns of sarcoplasmic reticulum on each side is known as triad. Together, the triad components make up a signaling apparatus converting repeated cell membrane depolarizations into spikes of free cytoplasmic calcium that trigger contraction. Now, let us see what is the mechanism of contraction. The contraction in all muscles is caused by sliding interaction of thick myosin filaments along thin actin filaments. The forces necessary for sliding are generated by other proteins affecting the weak interactions in the bridges between actin and myosin. During contraction, neither the thick nor thin filaments change their length. Contraction results as the overlapping thin and thick filaments of each sarcomere slide past one another. Contraction is induced when an action potential arrives at a synapse, that is the neuromuscular junction. It depolarizes the sarcolemma. The action potential is transmitted into the cell along the T-tubules to the terminal cisternae of sarcoplasmic reticulum to trigger calcium release. The calcium ions are released from terminal cisternae into the sarcoplasm. In a resting muscle, the myosin heads cannot bind G-actin because the binding sites are blocked by the troponin-tropomyosin complex on the F-actin filaments. Calcium ions released upon neural stimulation bind troponin, changing its shape and moving tropomyosin on the F-actin to expose the myosin binding active sites and allow cross bridges to form. Binding with actin produces a conformational change or pivot in the myosin. which pulls the thin filaments farther into the A band towards the M line. Energy for this pivot and pulling of actin is provided by hydrolysis of ATP bound to the myosin ends. ATP bound to the myosin undergoes hydrolysis via the enzyme myosin ATPase and loses a phosphate to become ADP. This allows the myosin end to change shape and pull on the actin, which is referred to as power stroke. Myosin pulling on the actin causes the muscle fiber to shorten. After this step, myosin binds to another ATP and detaches from the actin. In the continued presence of calcium and ATP, this attachment, pivot detachment events occur in a repeating cycle, each lasting about 50 milliseconds, which shortens the sarcomere and contracts the muscle. A single muscle contraction results from hundreds of these cycles. When the neural impulse stops and levels of free calcium diminish, tropomyosin again covers the myosin binding sites on actin and the filaments passively slide back and sarcomeres return to the relaxed length. In the absence of ATP, the actin myosin cross bridges become stable, which accounts for the rigidity of skeletal muscles called rigor mortis that occurs as mitochondrial activity stops after death. In the contracted stage of muscle, the following changes occur in the sarcomere. The length of the A band does not change. The length of the H band and the I band decreases and becomes short because of the sliding of actin filaments over the myosin filaments. The Z discs move close to each other, hence the total length of the sarcomere is shortened due to the above changes occurring. With this background, now let us see how the sections, that is longitudinal and transverse sections of a muscle, look like under the microscope. This is the diagrammatic representation of the longitudinal section of the skeletal muscle as observed under low power of the light microscope. The features are unbranched cylindrical fibers, peripherally placed multinuclei showing cross striations of alternating light I and dark A bands. The nuclei are placed just beneath the sarcolemma in the sarcoplasm. Each I band is seen to be bisected by a dark transverse line, the Z line. The sarcomere extends from one Z line to another Z line. Each muscle fiber is surrounded by endomysium with blood vessels and nerves in it. The ultrastructural features cannot be observed under light microscope. It is only observed under the electron microscope. So these are the features which can be observed under the light microscope. This is the photograph of longitudinal section of skeletal muscle under high power of microscope. You can pause the video and go through the features. This is again another photograph of longitudinal section of skeletal muscle under high power of the microscope. This is one more photograph of longitudinal section of the skeletal muscle under the high power of microscope. You can pause the video and see the features at your pace. This is the diagrammatic representation of the transverse section of the skeletal muscle as observed under low power of the light microscope. The features are: whole muscle is surrounded by connective tissue sheath called epimysium, which is not seen in this picture. It is made up of numerous muscle fascicles or bundles. Each muscle bundle is made of numerous muscle fibers and is surrounded by connective tissue sheath called perimysium. Each muscle fiber is surrounded by connective tissue sheath called endomysium. Each fiber at the level of cross-section will show one or two peripheral nuclei. Blood vessels and nerves are present in epimysium, perimysium and endomysium. So these are the features which can be observed under the light microscope. This is the photograph of transverse section of skeletal muscle under the low power of microscope. You can pause the video and go through the features. This is the photograph of transverse section of skeletal muscle under high power of microscope. With this, we have completed the histology of skeletal muscle. To recall, we have discussed the general introduction, types of muscle fibers, specific features of skeletal muscle, arrangement of connective tissue and muscle fibers, ultrastructure of muscle fibers, and how it is observed under the microscope with photographs and diagrammatic representations. In the next session, we will discuss on the histology of cardiac muscle and smooth muscle.